you know already what is F. That is F helix. And E is E helix. Now what are 8 and 7? That's the number of amino acid starting from 1 to this one, 8. So if a helix contains 12 amino acids, let's just say this 12 amino acids, number 8 amino acid is histidine. F8 histidine. And also this one, A7 histidine. At the E helix, the number 7 amino acid is histidine. Okay? All right. So where are they? Let's look at this. Wait a minute. Wait. Okay. So here you are. Okay, so here we are. This green thing here is the plane of your heme. And in the center is your iron. And this is attached. The fifth coordinate of your iron is your histidine with an imidazole ring here. And this is A7. A7 is not attached to the iron. It is just there, as to hinder just something here so that only oxygen can bind to iron. Okay. Now we have an element, a molecule which binds to iron stronger than oxygen. And this is your carbon monoxide. Now, in order that this will be prevented from binding to the iron, because carbon monoxide is a poison, this is the work of the A7. So it is just there as a barrier. So this type of bonding here, where it is not 90 degrees, which is 90 degrees, 90 degrees to the heme there, 90 degrees to the heme. So if this one is straightened up like that, the binding is 25,000 times stronger than the binding of oxygen to iron. So in order to prevent this, okay, the histidine E7 is right there, so that there will be no 90-degree attachment to the heme of your carbon monoxide. Okay? All right. Now, if E7 is not there, this oxygen here will just move this way and becomes a 90-degree angle, in which the binding is 25,000 times stronger than this one. So which means without this, you cannot go out into the street where there are plenty of vehicles running around, and it's the discharge from the vehicle has carbon monoxide, and you will just die there. However, if there's so much of carbon monoxide, a very high saturation of carbon monoxide, although the binding is not no, as strong as this one, the oxygen, because of this histidine here. Since there are so much of carbon monoxide, then all of those heme will be bound with carbon monoxide. So poisoning can occur if the concentration of the oxygen is very, very high. Okay? All right. So there you are. This one here. Oh, sorry. This one here. This is the natural way where your oxygen binds to your iron. Now this one requires that all the electrons will be in 180 degrees. Okay, as you can see, our oxygen, this is 100 around 120 degrees. Okay. All right. Now we'll compare the oxygen dissociation curve for myoglobin and hemoglobin. Now, if you look at this is your myoglobin. The myoglobin has what you call a hyperbolic curve, and the hemoglobin a sigmoidal curve. This is your percentage saturation. So from 0% to 100% saturation. Now here is your partial pressure of your oxygen. Okay. Now if you look at this, your myoglobin, even at a very low concentration of oxygen or partial pressure of oxygen, now this is 10 here. If you bring that up, it is already 70% saturated. No? So which means that your myoglobin easily is saturated by a very, even a low concentration of oxygen. This one, your hemoglobin, as you can look at it, at 10 partial pressure of oxygen, it's only up to here. While your myoglobin is already there, 70% na. Ang hemoglobin, it's only not even 10% saturation. Now this is because your myoglobin is only one subunit, only one polypeptide chain. Okay? Your hemoglobin is made up of 4 subunits. So which means each subunit has a heme molecule. So there are 4, so there will be 4 molecules of heme. And the heme molecules are not bound to oxygen at the same time. They are not bound to oxygen at the same time. So that the curve shows a sigmoidal curve. So there are 4 heme to be bound with oxygen. So the first binding is quite hard, as you can see this, and then the second binding becomes a little bit easier, and the third binding is easy enough, and the fourth is easy. So that the curve is like this, sigmoidal. Okay. This is characteristics of enzymes that are made up of more than one subunit. So 2 to 3, 3, 4, 2 subunits, 3 subunits, 4 subunits. You can say a dimer, a trimer, or a tetramer. Okay. So it says here, arterial oxygen tension is about 100 millimeters mercury. No? Mixed venous oxygen tension is about 40 millimeters mercury. This is taken from an active muscle or a muscle that is contracting. So there is using so much of oxygen, so it's only 40 millimeters mercury. And the minimum oxygen tension required for the cytochrome oxidase is about 5 millimeters mercury. So the enzymes requires only 5 millimeters mercury. So this is around here, between 0 and 10, that's about 5. Association of change into a tetrameric structure, hemoglobin results in much greater oxygen delivery than would be possible with a single chain or a single subunit. Okay. All right, we will explain that some more, no? So you'll understand. So the allosteric properties of enzymes is very similar. The allosteric properties of enzymes is very similar to the allosteric properties of hemoglobin, resulting from their quaternary structures. Quaternary structure, still remember our protein? We have 4 structures. Primary structure is made up of just amino acids string together by what you call peptide linkages. Then the secondary structure, it forms helices and beta pleated sheath. And then the tertiary is when the polypeptide chain having your helix and beta sheets and the bends and the loops. So it is a tridimensional form of the polypeptide. So that's tertiary. And quaternary structure is when there are more than one subunits of the protein. In this case, hemoglobin has 4 subunits, and we call them 2 alpha and 2 beta subunits. Okay. Allosteric means, what does allosteric means? It means another place. Properties of hemoglobin provide additional model also. We will go into that later on. Okay. Hemoglobin is tetrameric, meaning there are 4 subunits. And the 4 subunits, there are only of 2 types, alpha and beta. So you have 2 alpha and 2 beta. And this is your mature hemoglobin. Okay. So the normal hemoglobin is alpha 2, beta 2, meaning 2 alphas and 2 betas. That's hemoglobin A or normal hemoglobin. This one is fetal hemoglobin. 2 alphas. And what is this?
Gamma.
Gamma.
Gamma. Okay. 2 alphas and 2 gammas. This is fetal hemoglobin. Fetal hemoglobin. That is the hemoglobin that you have while you are still inside the womb of your mother. Okay. Now this one is 2 alpha and again a sickle cell beta. 2 also. This is one of the amino acid here.
Lagyan mo sa freezer yung ano, ilang bot na. Lagyan mo sa freezer.
I unmute na. This beta has a genetic disorder in which the number 6 carbon atom has been replaced. A normal carbon atom, the amino acid, I'm sorry. The normal amino acid at the number 6 position is glutamic acid. And in sickle cell anemia, it is replaced by valine. Now, if you look at the 2 amino acids, glutamic acid is base acidic amino acid. So if it is base acidic, it bears a negative charge. Okay? However, valine is a neutral amino acid. So there is no charge, 0 charge. Okay? So the 2 are not compatible, no, with each other. If let us say the glutamic acid at the number 6 position of a normal beta chain has been replaced by aspartic acid, then there would be no any what you call a disorder, because the amino acid that has been replaced is replaced by a similar functioning amino acid, since both glutamic acid and aspartic acid are acidic amino acids. So their function are not being replaced. Okay? All right. The primary structures of beta, gamma, and delta chains of human hemoglobin are highly conserved. So you have here 2 alphas and 2 deltas, which is known as hemoglobin A2, a minor adult hemoglobin. Okay? Hemoglobin. Okay. Where are the 4 globin subunits? Okay, they have different colors. This one is darker blue. That is one subunit. And this one here is blue, another subunit. And then one here is green, And then one where here is lighter green, some sort of a gray color. You see? And each of the subunit has a heme. This one here in red. Okay, another red structure here, which is heme. Another heme here for this subunit, and another heme here, this subunit. Okay, so what is this? This black thing. This black thing is a molecule produced in the glycolysis of glucose known as 2,3-BPG or 2,3-biphosphoglyceric acid or biphosphoglycerate. Okay, the one here. Now, what is its function? It is to stabilize the two beta subunits of your hemoglobin. Once they are there, the subunits of the hemoglobin are in a tense, very tense position or configuration. It has no freedom to move around when this is present. This is present only during deoxygenation, or meaning the oxygen from the heme are removed or being downloaded into the tissues that require oxygen. So which means it is now in a state of what we call deoxyhemoglobin without oxygen. And in order that, because once the oxygen are there, the 4 subunits becomes very loose. Okay? Once it unloads the oxygen, your 2,3-BPG comes around and make the 4 subunits or stabilize the 4 subunits from being relaxed or loose. So they become tense or taut, T-A-U-T. So they won't fall apart. Okay? Myoglobin and the beta subunits of hemoglobin share almost identical secondary and tertiary structures. Meaning to say that the beta subunits of hemoglobin is almost identical, to myoglobin. Okay. Despite the differences in kind and number of amino acids present in myoglobin, there are 153 amino acids, and the beta polypeptide of hemoglobin share almost identical secondary and tertiary structures. Similarities include location of heme and the helical regions. Remember, there are 8 helical regions. The presence of amino acids with similar properties at comparable locations. Although it possesses 7 rather than 8 helical regions, the alpha polypeptide of hemoglobin also closely resembles myoglobin. Your myoglobin has 8 helices. Your beta subunit of your hemoglobin has only 7. 7 for the beta subunit of hemoglobin, while there are 8 for the myoglobin helices. Hemoglobins can bind up to 4 molecules of oxygen per tetramer or per molecule of hemoglobin. 1 oxygen per heme. In addition, hemoglobin will bind a molecule of oxygen more readily if other oxygen molecules are already bound. This is what we call cooperative binding. No? So there are 4 binding sites. And we mentioned that if it is a deoxyhemoglobin, it is very tight because of the presence of 2,3-BPG. So how can an oxygen bind? So you have to have a higher concentration of oxygen. Which part of the body has a higher concentration of oxygen? And it's only in the lungs. So therefore, that hemoglobin has to go to the lungs in order that the hemoglobin can carry the oxygen. So there are 4. As we said, it has been mentioned here that oxygen binds readily if there are other oxygen molecules already bound. And we term that cooperative binding. So as we said, there are 4 binding sites. The first binding is very hard. Diba? Oh, boys. The first time, boys, mahirap talaga, no? Oh, walang yun.
Mahirap po talaga, Doc.
Mahirap talaga. And the second time, it's easier na. A little bit easier.
Yes, Doc.
Oh, yes.
Yes, Doc.
Oh, and the third time, lalo na. And the fourth, very, very easy. So that is what they call cooperative binding. We start with a very hard to bind, then becomes a little bit easy, and then easier, and then very easy. Huh? Okay. This phenomenon permits hemoglobin to maximize the quantity of oxygen loaded at a partial pressure of oxygen of the lungs and the quantity of oxygen released at partial pressure of oxygen of peripheral tissues. You know, the peripheral tissues always has a lower concentration of oxygen. All right. That's cooperative binding. That's very, very important. Never mind that. Oh, here we are. This is very crucial. You still remember this green thing? That is the plane of the heme. No? It's a porphyrin plane. Okay. Because those cyclic compounds there are 4 of them, the pyrrole rings. Those pyrrole rings known as porphyrin. Okay? That's the porphyrin ring, the plane of the porphyrin ring. And the oxygen, the iron is just a little above, no? As the equation is a little above although it is bound to the nitrogen here and the nitrogen here, so there are 4 nitrogens, okay? Now when oxygen comes in, look, when oxygen comes in, all right, the iron pulls, no, pulls this spiral ring down into the plane. Okay, very slight movement only. It's very, very small. You cannot even see that. But there is a movement from above, which is this is about 0.3 angstrom. This is pulled inside, as you can see it here, and there is a movement of 0.1 angstrom. Okay, that movement is very crucial. Why? Because look, this whole thing here is pulled. It's being pulled inside. Now, I wish I can. How can we remove this?
I-move ni mo kay ang cursor kay manaw.
Can you still see this? No. This is the oxygen here that is
Yes.
pulling it down.
Yes,
Okay.
Doc.
All right. That's the oxygen that is pulling it. So there is a movement, a very slight movement, but that slight movement is very crucial. Because once you move this, look at this. You have 2 alphas. Alpha 1, alpha 2. Beta 2, beta 1. Okay? That movement creates, okay? This will move. This will move this way. That will move that way. Okay? That way. This one will move this way. And there will be a 15-degree displacement. So there are 2 forms. This one is deoxygenated, or what we call T form, tense or taut form. Okay? Once oxygen comes in, there is a movement of 15 degrees, and we call that the R form or relax form. Okay? You see the difference now? So what is the implication of this movement? All right. Look at this. We'll start from here. There are 2 alphas and the 2 betas. They are joined together by salt bonds. Okay? Salt bonds. Another salt bond. Another salt bond. Another salt bond. There. Okay? All right. Now, when oxygen binds to the heme here, look at that. The salt bonds are being threatened already. Now, there's wavy line. It has not been broken yet. Huh? Now, this one is still very strong. Okay? Now, sometimes this will go directly, that all the bonds will just be broken. The salt bonds. Salt bonds are created by positive and negative amino acids. Okay? All right. So if you look at this, another oxygen binds to the second. So the bond here is already obliterated, threatening the bonds here. Straightening the bonds here. And then this can also completely go this way. It doesn't have even to go that way. No? So sometimes it just go this way. And only 2 oxygen yet. And all the bonds are broken. So the hemoglobin, the globin is relaxed. So oxygen can come in. Just like that. And oxygen can come in. Now look at this. There is still bonding here. No? Now once that is broken, then it is very easy for oxygen to occupy this. And so you have 2 oxygens now in the 4 subunits. The 2 alphas and the 2 betas. Again, through cooperative binding. Cooperative binding. Okay. So the transition between 2 structures is influenced by protons, carbon dioxide, chloride, and 2,3-BPG. Okay. Protons. When there is an increase of protons, will the pH go up or go down?
Up.
Huh?
Down, sorry. Down.
Down. The pH becomes
Down.
acidic, more acidic. Okay?
As-
All right. Now, so with carbon dioxide. So with chloride. And so with BPG. Okay. The higher the concentration, the higher the oxygen must be bound to trigger the transition, or meaning removal of the BPG when there are more of high oxygen tension. So fully oxygenated molecules in the T structure are now fully deoxygenated when it discharges the oxygen. And the R structure are not shown because they are unstable. Okay. Cooperative binding. All right. Your hemoglobin assists in the transport of carbon dioxide to the lungs. Now, carbon dioxide, where is carbon dioxide produced? What tissue?
Inside cells.
Inside where? Inside which tissue? Which tissue?
May
Astaman. Sige lang, sige lang. Ah?
Ace.
Well, you know, cells that are respiring. You know what is respiring?
Alveoli.
Ah?
skeletal muscle. Skeletal tissues.
All right.
Alveoli.
In your kindergarten, we were told there are 2 kinds of respiration. External and internal.
Internal.
Ah, okay. Now, external is the one that you can see. You are breathing. That's external. No? The internal is the utilization of oxygen. So where is the oxygen utilized?
In cells
In the
that
cells. Oh,
respire.
respiring cells. Respiring. Respiring cells. In the
Respiring cells.
through the respiratory chain. Diba? What is the other name of respiratory chain? Electron transport chain.
Transporting.
No, electron transport chain or respiratory chain. That is the internal respiration, meaning the oxygen is being used, no, by the cells, and carbon dioxide is formed. And this carbon dioxide has to be carried to the lungs to be expelled. Okay? Now, not all of the carbon dioxide formed in the respiring cells are carried by the hemoglobin. Not all, just only a fraction. It is not usually carried all by the hemoglobin. Okay? All right. As the blood goes into the lungs, part of those carbon dioxide that is formed from the respiring cells are expelled out so that the lungs can exhale it. Now, part of the whole parts of the carbon dioxide are in the plasma. And then through your very important enzyme, okay, this one here, carbonic anhydrase, no? Converts carbon dioxide and water in the plasma into carbonic acid. And once it reaches the lungs, spontaneous dissociation into bicarbonate and protons. Okay? This is carbonic anhydrase. A very important enzyme, both in the cells and in the lungs. Okay, so this is your Bohr effect. All right. Now we start here with carbon dioxide from what particular cycle? Krebs cycle. Okay, producing carbon dioxide, and carbon dioxide reacts with water through carbonic anhydrase. It becomes bicarbonate, no? Bicarbonate, and then this dissociates into protons and bicarbonates. So carbonic acid becomes bicarbonate and protons. Okay, it is buffered because if it is not buffered, there are plenty of protons here that is formed, and the plasma, no, of the blood becomes acidic, and we do not want that. So the hemoglobin buffers it. So what are buffers? You still remember?
They control the pH in the body.
Okay. So buffers, no, maintain the pH of the solution where they are being put into. Your hemoglobin is a very good buffer. And this hemoglobin buffers this hydrogen. All right. And when it is in the lungs already, the oxygen in the lungs, all right, and the buffered hydrogens from the hemoglobin exchanges. The hemoglobin takes up the oxygen and becomes oxygenated hemoglobin. And the hydrogen here carried by the hemoglobin is now thrown into and the bicarbonates that are formed here, which is also carried by the plasma, now forms again carbonic acid, and then the carbonic acid carbonic anhydrase in the lungs convert the carbonic acid into water and carbon dioxide, which you exhale. No? So the water there is this moisture every time you breathe out. No? You are breathing out water and carbon dioxide. Now, if you do not believe that there is water that forms part of the air that you breathe out, you just have a cold surface, or let's say a mirror that is cold, and then you breathe there, and you will see the moisture. No? Okay. So carbon dioxide is expelled out. And this is what we call the Bohr effect. The Bohr effect. Carbon dioxide generated at the peripheral tissues meaning the respiring cells, combines with water to form carbonic acid, which dissociates into protons and bicarbonates, as shown here, protons and bicarbonates. Deoxyhemoglobin acts as a buffer by the protons and delivering them to the lungs. And in the lungs, the uptake of oxygen by hemoglobin releases the proton that combines with the bicarbonate ion, forming carbonic acid, which when dehydrated by carbonic anhydrase becomes carbon dioxide and water, which is exhaled. There you are. That is the Bohr effect. Okay. All right. Protons arise from rupture of salt bridges when oxygen binds. You remember that? Atong cooperative binding, the salt bridges are being broken. As the salt bridges are being broken, protons are released. Okay. The protons are released after rupture of salt bridges. The Bohr effect is dependent on the cooperative interactions among the subunits. That is cooperative binding. Associated heme groups of the hemoglobin. In the lungs, the partial pressure of oxygen drives the conversion of hemoglobin from a tense or taut to a relaxed state, meaning oxygen has already been bound to the heme, breaking the salt bridges involving the beta chain residue histidine 146 and subsequent dissociation of protons from histidine 146 drives the conversion of bicarbonate to carbonic acid, figure 611. Now, remember, histidine is a basic amino acid. So it is positively charged. So if there is positivity, there is protons. And when released, protons are positively charged. Okay? Now, in respiring tissues, high concentration of CO2 drives the 148 formation of carbonic acid and the acidification of your red blood cells. Upon the release of oxygen, the T structure and its salt bridges reform. This conformational change increases the pKa of the beta chain and histidine 146 residues, which bind protons by stabilizing the formation of this state. Hemoglobin, the carbon dioxide driven increase in proton concentration enhances the release of oxygen. So this is what happens in the respiring tissue, where there is high CO2, which is responsible for kicking out the oxygen from the heme so that it can go to the tissue. high carbon dioxide tension, high protons will increase the release of oxygen in the tissues. Okay? All right, this is the BPG. This is the molecule of BPG. This is a very unique compound because this is only formed by the red blood cell, no, when they utilize glucose through anaerobic glycolysis. When we say anaerobic, it does not use oxygen, no? It does not use oxygen, anaerobic. Why should a carrier of oxygen, which is the red blood cell, cannot perform aerobic glycolysis? When it has plenty of oxygen? Wala.
Because it has no
Kasi-
mitochondria.
Very good. Because it does not have the ability to utilize oxygen. The utilization of oxygen in cells, it is because of the presence of mitochondria. And the mitochondria, the inner mitochondrial membrane, has what you call your respiratory chain, which is a chain of enzymes, which transports electrons from one enzyme to another until the final acceptor, which is oxygen, forms water. So that's how it is. So even though they carry so many oxygen, they cannot use it because they do not have the apparatus to use it. Now you remember, I guess you have already some idea of how your red blood cells was formed. Now the red blood cells was formed when it is immature. It contains all the organelles. What do you mean by organelles of the cell? What are the organelles?
Parts of the cell.
Oh, organelles. So organs that are small.
Parts of the cell that have
Okay,
function.
yeah. All right, so you have your mitochondria, Golgi apparatus, di ba? Lysosomes, all of that, all cells, no? Now, when the RBC is being formed, it all has the organelles. But as it matures, all the organelles are removed. Why should all those organelles, which is very important for the life of the cell, why are they removed? In order that hemoglobin can be accommodated. Approximately, there are 240 hemoglobin molecules per red blood cell, per RBC. So you can just imagine how many hemoglobin per RBC. That's why it has to have space, and all of the organelles had been removed. So how does the RBC survive? It survives by utilizing the glucose, which is the source of its energy, through the enzymes still present in the cytoplasm of the RBC. These are the glycolytic enzymes. And since it cannot use oxygen, type of oxidation of glucose is anaerobic glycolysis. It is a very expensive way of producing oxygen. Producing energy. Why? Because per molecule of glucose, only 2 ATPs. You know what is an ATP, no? 2 ATPs are only formed. It's a very expensive way because you need to have so many glucose, so much glucose, and you only produce 2 ATP. Okay? So, when the enzymes of your glycolysis, after forming so many enzymatic reactions, they become old and tired, and they cannot perform its function anymore. So what happens? The RBC also dies. So what is the average lifespan of an RBC?
125, 120 days.
Okay, see? You can see that they easily dies within that period of time because the enzymes are already tired and worn out. And where are they going to be destroyed? The RBC. All right, it's spleen.
Spleen.
So as this go to the spleen, they are destroyed there. The splenic tissue is just like a filter. Small holes, very, very small holes. But a healthy RBC can squeeze into the holes. So it passes through, healthy. But the battered RBC, when the membranes are already battered, as they squeeze through the hole, they rupture. And once they rupture, they die there. That's why you call the spleen the graveyard of the RBC. You get it? You connect the dots already? Huh? All right, so here is the BPG, which is formed only by the RBC. And its function is just to stabilize the 2 beta subunit of your hemoglobin. Okay, this amino acids on this side belongs to 1 beta subunit, and this amino acid on this side belongs to the other beta subunit. And these are the amino acids that interact with your BPG so that the structure becomes stronger and it is not loose, it is not relaxed. So this only happens when the hemoglobin discharges all of the oxygen, and which is called a deoxyhemoglobin. And BPG comes in to stabilize the 2 beta structures so that the hemoglobin will not fall apart. Get the idea? So this is what is the function of that. No? Okay. Adaptation to high altitude. Okay. Physiologic changes that accompany prolonged exposure to high altitude. What is in the high altitude? Do you have high concentration of oxygen or less concentration of oxygen?
Less
Less
concentration.
less
Alright.
consideration.
Less concentration.
Okay, less. So if it is less concentration, there is less oxygen that goes into the lungs. Diba? So, the deoxyhemoglobin will have a hard time getting oxygen because the concentration there is low. You need to have a high concentration in the lungs. Okay? And as you can see here, BPG lowers the affinity of hemoglobin for oxygen. You have seen that, the deoxyhemoglobin, because it makes the hemoglobin very tight. All right. So what is the remedy? What is the physiologic remedy of this? When you are staying at a high altitude or on a mountain, the physiologic remedy would be
Increase the number of
Increase the number of
erythrocytes.
erythrocytes. Okay, that's it. So that's why you will have an increase production of RBC. Diba? RBC. So those people who are living in the high mountains or high altitudes, they have an increase of RBC. No? Increase of RBC. So that when they go down to the lowlands and there's plenty of oxygen, all of this hemoglobin are now being bound with oxygen. And if they are fair skin, you will see that the face becomes reddish or pinkish. It's because there's so much of red blood cells, plenty of hemoglobin to carry oxygen. That is the result. Okay. You know that already, no? All right, now what we have here are mutations of hemoglobin. Once we have hemoglobin M, or and then hemoglobin S. What is very common is hemoglobin S, or sickle cell anemia. Now, by the way, I forgot anemia. What's the opposite of anemia?
Polycythemia vera.
Very good, very good. Polycythemia. That's what you get when you are in the high mountain, staying there for a long time. Polycythemia. Okay. So, more common is this one, hemoglobin S, or sickle cell anemia. Yeah? This is how it is. Polymerization of your hemoglobin. Polymerization. So you'll notice that all your hemoglobin are being attracted to each other. Magkumpul-kumpul na sila. A very good way to spread the COVID virus, no? Because the population now are all together. Now, what really happened here is this. the mutation is on the beta subunit. The number 6 amino acid of the beta subunit, which is normally occupied by glutamic acid, has been replaced, genetically replaced by valine. Okay. And as a result, and as a result, whenever the hemoglobin, which is full of oxygen, which we call an hemoglobin, when it is unloading, it unloads the oxygen to oxygen-starved tissue, the result is deoxyhemoglobin. And it forms what we call a sticky patch. Sticky patch. So, normal hemoglobin, no? This one is oxyhemoglobin, meaning there is plenty of oxygen. Once it removes the oxygen, it becomes a deoxyhemoglobin, S. Okay. And you form a sticky patch. Sticky patch. All right. And the normal hemoglobin, it has a complement there, the beta subunit there. Okay. So a normal a normal hemoglobin A and a deoxyhemoglobin S can patch here because that is a sticky patch and this is the complement. That one there is a complement. Then it is now attached to it because that is sticky. And the other like this because there's a complement on this side. So other hemoglobin S will just attach here. And so that's what happens now. They're attached to each other. Okay. And once this is seen in the microscope, that they usually are found in the inner membrane, no, in the inner side of the RBC cellular membrane and form a crescent moon. And they become hardened. All right. Now, once this passes through your spleen, and once it passes through the spleen, the RBC tries to go inside a small hole because it can, a normal RBC can easily pass through. But an RBC with already a hemoglobin S that are being formed and hardened, then that would stick out of the membrane and the membrane breaks. Hemolysis occurs. So RBC are destroyed, producing anemia just as a result of that. No? Okay. There are plenty of this, no? Africans are usually in, they have this gene of your hemoglobin beta subunit, where there is a replacement of the glutamic acid to valine. Okay. Okay. What is glycated hemoglobin or hemoglobin A1C? Glycated meaning your hemoglobin is decorated with glucose. When can this occur?
Diabetes.
All right. When you have a high sugar content in your blood, you may not have the signs and symptoms yet of diabetes mellitus, but your hemoglobin is already decorated with glucose. The glucose attaches itself to the protein, and that is the globin of your hemoglobin. Okay? So blood glucose enters the erythrocytes and form covalent bonds. The amino groups of lysine. Okay? And so your glucose is there. Now, if you do not have the signs and symptoms yet of diabetes, and when they do an examination of your RBC and your hemoglobin, then if they say hemoglobin A1C, that your hemoglobin is decorated with glucose, then that is a sign already that you will have diabetes mellitus, a frank diabetes mellitus. So this is an early sign that you'll be having diabetes mellitus. Now, we have already a high incidence of diabetes mellitus in the Philippines. No? Does mean that Filipinos are eating well. It just means that there are two kinds of diabetes mellitus, if you still remember. Diba? Adult
Type
onset
1,
type.
type 2.
Type 1 and adult onset
Type
is
2.
type 2.
Type 2.
Oh. And genetic disorder
Type
Type
1.
is
1.
type
Type
1.
1.
Type
Okay.
1.
Okay. Type 1 is the inability of your pancreas to secrete
Produce
insulin.
insulin.
Oh. But type 2,
Insulin.
it does not mean that you do not have insulin. You have plenty of insulin. But what is the problem
Insulin
is,
resistance.
insulin
Insulin
resistance.
resistance.
All right. The problem there is that your cells in insulin resistant does not have a receptor for insulin. Now remember that your insulin cannot get inside the cell unless they bind to a receptor. Insulin is water-soluble hormone. And water-soluble hormones or water-soluble compounds cannot pass through cellular membrane. Cellular membranes are lipid bilayer, meaning a fatty tissue, fatty, oily molecule, and water cannot pass through it. Uy, thank you naman. Sorry.